Hello, hello, hello. Yep, there we go.
Perfect. Awesome. Thank you very much. And have you guys been having a mic running?
Yes.
No?
Thank you so much. Yeah. Except, I think it's just me and Nataša though. Oh, okay. So, it's gonna be charm coming out the wazoo.
I cannot wait.
We're just so lively. Yeah.
Oh, okay.
Amazing. I don't think so, but it's great that you're here. It makes it so much easier.
When
Well,
you
you
come
are.
out,
It's very
your
important.
slides will, they'll all be in the folder, so just find. Double click and you're good to go. And then full screen.
We've got you, if you don't. We'll be here to help you out along the way, don't worry. Yeah. Yeah, I'm one of the senior members on the biogEosciences team. So yeah. Super fun. Just to do, because we only have the ECR section today. No, it's been a long time thing. Yeah, I think it's been like that for some time. But thank you for the compliment, thinking that I could make something so bad. Flattering. Yeah. Yeah, I presented Wednesday though. Yeah. Well, I'm doing my PhD in Guelph, but I'm presenting on my master's work in Waterloo, and that is on like wildfire recovery strategies in peatlands. Yeah. Yeah. I guess I'll find out. Are you presenting in this session as well? I'll find out. I don't want to spoil the surprise. Well, I'll try to take it. Good to have a little. Yeah, I mean, I like the. Oh, you should tell her she was very worried and asked her questions. Yes, so like it's showing.
Yeah, is this okay? Oh, is there stairs? Okay, so like come on my phone to me. And then I don't know. Do you want to run a question? Yeah, I might. I mean, I can also.
So very tiny. Okay, so when they share, how do you do it? Hit the share screen button.
Yep.
Okay, so
And maybe.
here on click on window and pick just this one. Because we want to take the slide first. Yeah. Okay. Just that option.
Yeah.
And so that's why you're doing it. Yeah. Now, and let's see if it's on the lobby. 302 right? The other thing is when you're searching, you have to have it on all, or else have the specific date. Oh, so you need to go back over there.
What do I have up here for, do I have a clicker?
I think we got a clicker. Test, test. I think it's good. Oh, is there a lag? Hmm. Oh, that is aggressive. I mean, fair. We're going to get started in a few minutes. We just have a few technical difficulties. Snap, snap, snap. Good to go? All right, let's get started. Welcome back to the afternoon session, part 2 of our biogeosciences ECR session. I'm really glad to be running this session again this year. I think this is our 3rd year in a row that we've kind of had a biogeosciences section to highlight the work, the really incredible work being done by our ECRs. And so thanks for coming out to support them. There's a lot of really great science being shown in the session, and I'm not just saying that because I'm in this session. This is dangerous. Who gave me a microphone? So we're going to start things off with a talk from Dr. Graham Clark. Graham did his PhD in reclamation research out in Fort McMurray, and now he is launching a brand new, very exciting research program looking at carbon sequestration in mudflats. So let's give a round of applause to Graham, and he's gonna give us. He's our invited speaker, so he's gonna give us a little bit of a longer talk.
Does this work? Okay. I'm just going to set up a timer here. Right. So yeah, I'm here to talk to you about a new research program I have started up. So there's not a lot of results, but I will go over our preliminary results from last season. And it's in the mudflats of the Bay of Fundy of New Brunswick. And so as Nataša mentioned, my research career up to this point has been in reclamation, trying to rebuild environments and how that relates to carbon and water cycling. And so now I'm moving east. One of the big things I was looking at was areas I could focus. And so I want to start by thanking a whole lot of people, but really I'm going to focus on the HQP because they do most of the hard work. They're out there every day collecting data for us. So that's Eke, Demeath, Courtney, Abigail, and Naman. They're all underneath the MyTax there. Let's see if this laser works. There we go. Underneath MyTax there. And of course, our research partners, Net Zero Atlantic, Ducks Unlimited, some funding coming from NSERC, and our partners at Environment Climate Change Canada and Rewrite, which is a Horizons Europe project, who are doing almost a parallel study to what we're doing here over in France. So looking at mudflats there and spanning the Atlantic and trying to compare results. Right, so when I got to St. FX, I basically started cold calling all the researchers I found who were doing anything at all that I found interesting and related to research projects and questions that I was into. And I found that quickly that unlike when you're in Western Canada and everyone's talking peatlands, everybody's talking coastal out west, or out east, which makes sense. There's a lot of coastline there, and there's a lot of coastal problems. There's erosion, there's loss of habitats, there's sea level rise, there's storm surges, there's all sorts of things that are problems, and coastal restoration is touted as a major solution to that. And so as a carbon scientist, my big angle is being able to approach organizations like Ducks Unlimited Canada and talking about measurement, validation, reporting. So when they restore a chunk of the coastline, they can't count the carbon credits and access carbon markets right now because of a lot of the uncertainties I'm going to get into in this talk. And so a lot of my research program is trying to understand the science better, particularly something called lateral transport, so that we can better inform these MRV processes so that organizations like Ducks Unlimited can come in and restore coastline and help offset the cost of that by accessing carbon markets. So for those of you who are familiar, I'm sorry, this might be review, but blue carbon is this buzzword used to describe carbon accumulating ecosystems that are either tidal or near shore ecosystems. So most of the work is focused on marshes and mangroves, and there's been a ballooning work looking at seagrasses. And so this paper by McCready et al. In 2021 really highlights all these different ecosystems and how we might be able to leverage them for natural climate solutions. And the big research gap that they highlight is this thing called lateral carbon. So if vertical carbon exchange is things like photosynthesis and respiration, lateral carbon exchange would be things like the tide coming in and washing away all those sediments that have accumulated the carbon. And so we don't know what's going on laterally. And so because of that, investors aren't very interested in saying, yes, we can guarantee the carbon's being sunk here. So we're trying to figure out this lateral question of where's the carbon going? Where's it being fixed? How is it being fixed? And can we say for certain that the carbon's going to stay there? One research gap, however, that nobody's really picked up on is these ecosystems kind of right here on the edge and this one right here. These are called tidal flats. And in a lot of places of the world, the difference between the vegetation and the low tide line is very short. But there are places around the world, such as the Bay of Fundy, where they can be hundreds of meters, if not kilometers wide. And so these are tidal flats. In particular, these are mud flats, and we classify them by their sediment size. And so this is about 800 meters. This one's Peck's Cove. And you can see the drainage patterns on them. So as the tide pulls out, they form their own channels and their own drainage systems like mini watersheds. And that's important because I'll come back to that later in the talk. And so these tidal flats are incredibly important in diverse ecosystems. They, like I mentioned, they dissipate storm energy. They rise up with sea level rise to maintain the coastline. They're incredibly productive, and the algae that grow on them, we call it a biofilm, develops. The invertebrates come along, start feeding on that. The shorebirds come along, start eating that. It's a very critical habitat for migratory species. So I got my little piping plover up there. Because this is a pretty complicated ecosystem for something that just looks like a whole bunch of bare mud. We don't know a lot about these systems because they're very understudied. So for anyone in the room who isn't entirely sure where New Brunswick is, it's kind of the forgotten province, south of Quebec, north of Nova Scotia, east of Maine, and sitting right there, highlighted it for you. So everything I'm going to talk about today comes from the upper Cumberland Basin, which is kind of, if you think of the Bay of Fundy, splits the left or the more northern side, goes up to Moncton and Amherst, Nova Scotia, and so that is where most of our research is. It's easily accessible. Mount Allison University is right here, and so it creates a great spot for us to store our equipment. So a lot of our sites are there, but part of this program is expanding the observation network, and we're growing our sites every year. And then the Northumberland Basin is the stretch of water that separates Prince Edward Island from the mainland. And so we have sites out there to compare and contrast. So in the Fundy, you have these macro tidal systems, right? 12-meter tides where we're working, huge amounts of water coming in, leaving. With that, there's a ton of sediment. In the micro tidal systems, you still get these exposed flats, but they are only exposed at the lowest of low tides, and they're not nearly as productive. And we'll get into a bit of comparison with them. So here on the right is a map produced by Tim Webster and his research group at the Applied Geomatics Research from NSCC, and they kind of did a first pass of trying to map these ecosystems. And they're very hard ecosystems to map because if you're doing it from satellite-based imagery, you got to make sure your satellites are absolutely at low tide, right? These systems disappear as soon as the tide starts coming up. And one important thing in this area that we're researching is you can see these orange areas are the mud flats, and they make up about half of the coastal area. Like in terms of the coastal ecosystems, it's a large proportion of the landscape are these flats that are yet undescribed and uninventoried by Canadian carbon reporting or anything like that. They're just not there. They don't exist. So again, this is another image. This is Peck's Cove. This is one of the bigger flats. This is almost 2 kilometers wide at its widest point. And it looks exactly as it sounds, mud flat. It doesn't look productive. It's a really cool ecosystem to walk around in. You're out there working and the plovers all just kind of like run around you. They're like migrating their way across, and they just make this big bubble to try and avoid the humans. But mostly it's really neat, and wander around. It's a dangerous ecosystem to work in. You're talking upwards of 12 meters of tides. If you get stuck in the mud, that will kill you quickly. So you need to have good safety protocols, good buddy systems, all sorts of, a safety plan is essential here. Because not all this mud is, some of this, particularly in here, you're going to get stuck in a lot more than other parts. So how much does this matter in terms of carbon? And so the answer is we don't know for sure, but we know that salt marshes, which are the ecosystems just a lot higher on the intertidal zone, tend to accumulate a lot of carbon in their sediment. And so our preliminary, this is some cores from last summer. We haven't worked them up totally, but the first one we have run is about 20 kilograms in the top meter of sediment. And so that's on par with something like a peatland as opposed to a forest, which is roughly an order of magnitude lower in terms of how much carbon these ecosystems are sinking into those sediments. So they are very organic rich. And this is Abby here. She's starting her master's this year to really expand our network of cores in the mudflats. So we have about 800 cores in the marshes around this area, but we have almost none in the mudflats. So this campaign this summer is all about the mudflats. Another thing you can do with the mudflat, with the core data, is you can look at isotopic data to get an idea of where that carbon might be coming from. And so we know this little thing is over top of my graphic. I don't know if you can get rid of that, but it doesn't really matter. I can tell you about it. At about -20 here, that's an indication that the carbon is coming from the microalgae, so that biofilm that's growing. And at -13, that's more from the marshes. And so when we look at the Northumberland Strait, that like microtidal system over by PEI, we see a lot more influence of the marsh. And so there's a transport mechanism occurring that's moving carbon from the marsh into these flats. So if you're trying to restore the marshlands, some of the carbon that you're saying that you're fixing into that ecosystem might actually end up down in these mud flats. And so this is an important thing to consider because it's all connected in these, because the tidal energy can redistribute a lot of this. Whereas in the mud flat, we have much more, it looks like it's coming from the biofilm. So we know there's lots of carbon and we know it's probably a lot of it's being generated in sight. But that does not tell us the flux, right? And so I'm a flux scientist. I work a lot with systems like eddy covariance. I want to understand how carbon is moving around. And so that's where a lot of my research questions come in. Is this something that we can measure very easily just for a month of the year and extrapolate to know how much carbon is being fixed for the next 10 years? Or is it the type of thing that totally gets redistributed as soon as a hurricane comes along and changes everything? Or this area is very prone to ice flows, ice jams. So big icebergs get washed up on shore, they get washed away, they move around with the wind, they'll flatten any equipment you have out there, but they also freeze to the sediment and lift the sediment up and relocate the sediment kilometers downshore. So before you can get into lateral fluxes, we need to have that vertical flux pinned down. And so there's been a lot of work on the salt marshes, and I'll get into that in the future work. There's going to be a lot more. But the mud flats, we have no data. Nobody's really even dropped chambers on them yet to know how much what the vertical flux of photosynthetic uptake is for these. And there's recently been some work that shows maybe the ocean, when it floods over these ecosystems, it continues to be a sink of carbon. There might be a mechanism. There was a great paper last year by María et al. Out of France that showed that when the system floods, that there's still a definite signal of carbon uptake. And so these are the research questions I'm going to be spending a lot of this summer looking at before we can really get into the lateral. And so how do we do that? Well, chambers, everyone loves chambers, eddy covariance systems, but we're also embedding this study design into this hierarchical system. And so I'll start with the chambers. So this is fresh data. That's one of the reasons why it's a total mess if you look at it. This came out last week. My master's student got back last week. Demetrius brought it back and he quickly worked it up for me and he showed me that. And you can see that there's kind of a dial trend here. So on the right, you have the hour of the day, and on the left, you have time since emergence. So I'll walk through what he does very quickly. We walk out there, you drop down a stake at the water level as soon as the mud flat is exposed. And you have your partner drop out the stake maybe 50 meters away. So now you have a gradient of elevation, and you know exactly where you are. So you can use that as a proxy of time since the tide has exposed the mud flat. And you just walk between those stakes and take chamber measurements until the tide comes back in and he chases you off the mud flat. And so you do that again and again and again. He does it over a 2-week period so he can get the whole migration of the tidal cycle, right? It migrates roughly an hour a day. So you can, you get out there at the start of the exposure at 9 o'clock on Monday. Well, on Tuesday, then it's going to be 10 o'clock. On Thursday, it'll be 12 o'clock. And so you can slowly try and untangle the influence of things like PAR and temperature, the dial trends that we all know in terrestrial ecosystems, from this migratory issue that we're dealing with. The diatoms actually migrate down into the mud when the tide comes in, and they migrate back up afterwards. And what is that lag, and how are we going to account for that when we're trying to understand the carbon impacts? I should have said, but hopefully it's obvious by my little arrow there, that more negative means more carbon into the ecosystem. I'm a micrometeorologist, so old habits die hard. Negative means carbon's coming out of the atmosphere. The nice thing about chamber measurements, though, is you can couple them with things like genetic samples. So we can get the community composition right in the chamber when we measure. We can get chlorophyll, like pigment analysis, which is a lot cheaper, so we do that more frequently. We do the genetics only intermittently. And so you start to build up what communities are driving, what type of response you're seeing, and that light response in that time since submersion response. But you also, every single time we do a chamber measurement, we get the hyperspectral data of the reflectance. The genetic data will give us what's in the top 2 millimeters of sediment, but the hyperspectral data tells you what the light reflecting off the surface is, and from that you can tell what pigments there are, right? So what chlorophyll, that sort of thing. We know there's a decent relationship between certain indices, so NDVI is a popular one, and the chlorophyll content of the mudflats. And so this is work by Naman, the postdoctoral fellow who really drives a lot of, the PDF. He's a PhD student, and he's really driving a lot of the synchronizing these studies to make sure that we can scale them up. Because the next step is to go to drones. And so while we're out there, they're flying the drones in the same day. We fly the drones maybe 3 or 4 times over that 2-week period. And so from that, we can link that hyperspectral data to the drone data and scale up the carbon data. And then those drones are specifically picked, some of those drone dates, to be on the same days as the Sentinel overpasses. And so we can then scale up and measure the entire region, hopefully cloud-free. You're trying to get cloud-free days, you're trying to get low tide, you're trying to get a satellite to line up with your timing. It's a hard thing to synchronize, but last year we got a couple good days, and we're hoping for a lot more this summer. And so then that brings us to the eddy covariance system. And so eddy covariance in this environment is very hard to work with. So our towers have to be tall enough to stay above the high tide mark. You can see there's my battery box there. We're getting pretty close. If that was a wavier day, it might be big problems. And in fact, I put it deeper this year. So I'm about 1 meter deeper. And now my battery box is up here, and my AC system's a little higher to try and increase the footprint a little bit. But in the spring, the ice flows are going to smash anything you have out there to pieces. And in the fall, the hurricanes are going to destroy anything you have out there. So you really have to be go time, go and deploy. And so this structure needs to be assembled and disassembled very quickly when we need it, which has been proven to be interesting challenges. And we've been working on different designs to try and make that more effective, because we have more eddy covariance systems coming out this year. So just quickly, this is only from 6 weeks of data. This is by no means the full season, but this was last fall. This is just a quick wind partitioning. So eddy covariance, for anyone who's unfamiliar with the technology, you get a continuous measurement of the CO2 exchange, but it's based on some footprint. So which way the wind is blowing. If the wind is blowing from the north, you're measuring the ecosystem that's under that. And you can figure out exactly where that ecosystem extent is for every time period you want, and you can kind of triangulate and separate out different ecosystems. And so here we can see that the marsh is the biggest uptake, the black is the biggest uptake, but it also has the highest rates of respiration. So the total dial CO2 accumulation, total daily amount of carbon exchange is actually pretty similar because the respiration is so suppressed in the mudflat. And we see no evidence, at least in the fall, of that ocean uptake that they saw in France. That line is pretty flat, around 0. So that's good, because our water is very turbid. It would be hard to explain how photosynthesis was going on inside the Bay of Fundy. This is Courtney Allen's postdoctoral work. She's looking at modeling the actual migration of those diatoms up and down through the mud column. So the blue bars here are the tidal periods, and the green bars are either where the center of mass is relative to the surface or where the surface concentration is. And that surface concentration is really the key to linking all this stuff together, because that surface concentration relates directly to the chlorophyll, to the spectral signature, which we can use to scale up to things like satellite level. We can get the whole ecosystem, and we can tie it to the carbon. And so this is all bringing it all together. So going forward, as I mentioned, I'm getting another covariance system out there, and we have funding in. Hopefully we'll stick a methane sensor on it. Right now, we don't have a lot of belief that there's methane being produced in the mudflat, but because we're doing the genetic work, we should be able to see if there's methanotrophs, definitely if there's methanogens, there's probably some methane. We see bubbles when you're walking around out there. They're really reduced conditions, but the sulfur is very high too. So you don't expect a lot of methane, but you do get methane from the high marshes, so we're going to try and help separate that out. Right now, Sarah Knox and I are looking to hire a PhD student who will take all these East Coast soft sediment coastline towers and combine them with her West Coast soft sediment towers and start looking at the different drivers of carbon exchange between the two different coastlines, because Canada in general doesn't have a lot of coast flux data. Sarah's been great in generating BC data, but we have nothing for the Atlantic so far. So this will be, and then we'll be feeding that data into the Peppermint T model, which is developed mostly for American coastlines, but hopefully works well in Canada, but we'll see. Another project I have going from here is looking at inventories with Environment Climate Change Canada, so the carbon reporting. So Corinne Millard at Carleton, she's going to be doing the spatial mapping of a lot of these ecosystems a lot more thoroughly than these couple first passes. Not that didn't do a good job, it was great work, but looking at more thorough ways to map out these ecosystems. And then I'll be coming along and measuring the carbon in those different landscape units to try and upscale to the whole coastline of New Brunswick. And finally, we get to the part that I think is the most exciting work, is the lateral transport. And so this is the real big question, is how much carbon is washing away to the ocean? How much of the ocean carbon is washing into these systems and ending up buried? We just don't know. And so this summer, we have a master's student who's going to come in and take it a first pass at just measuring a whole bunch of cool stuff. So we're going to do a bunch of Lauren Summers from Dal. We'll be doing a bunch of paired wells and seepage meters. We're going to we're going to stick a weir in one of those drainage channels so that we can capture the drainage off the mudflat. We might put some vertical profilers in, so you can have chambers that are lined up basically at the covariance tower, and at high tide they all open, capture the tidal water, and then they all close. We could do something like an auto sampler we've been talking about potentially to get the time series of how the carbon changes in the tide. Because there's been a lot of work that shows upwards of 30% of the carbon is lost in DIC alone from these landscapes, in these coastal landscapes. And so that is tropical mangroves, so the odds of that occurring here is probably smaller, but we just don't know. We have no idea how much carbon is being moved around by this tidal energy. On top of that, the tide acts as a giant pump on the landscape. So it's changing how you have groundwater percolating out and interacting with the ocean, because you have this huge amount of volume of water amassing over the surface every 6 hours, right? 12 meters of water is a big weight on top of these systems. So with that, that's where I'm going with this project, and that's that. I don't know if anyone has any questions or follow up, but yeah. So, thanks. Got him petered out there.
Yeah, thanks, Graham. That was really great, and I think it's a very exciting project, and like looking forward to the next several years of CGU and you guys showing some of the results from this stuff. Does anybody have questions for Graham? Okay, we're gonna run it, and then I also have a question.
Thank you. Thank you, Graham. It was an extremely interesting project, and I wish you all the best. Quick question. You've presented the stocks for the first meter, which is a great indicator, but it doesn't really tell us how much carbon is sank. So why not do carbon sequestration rates?
Well, I mean, that's where we're going, right? That's what
Okay.
we're trying to get at, is like so we know the like you can carbon date the bottom of the core and figure out how much of that carbon over time, but then knowing how much is being fixed will also help us figure out the sources,
Absolutely, yeah.
right?
No, I had a small study in James Bay on the coast, and so what we found out on an uplifting coast is that the carbon sequestration rates in the mudflat and initial terrestrialization was 6 times higher than, and I don't want to offend any peatland people here, but it, so compared to the peatland or the peat material, carbon sequestration rates in the tidal flat was 6 times higher. So it is, when you said initially that 20 kilograms of carbon in a meter of sediment, what really matters is how much per year is sequestered to really draw that conclusion that it is like peatlands. I think it's much higher than peatlands.
Certainly, but it depends on what you're looking for, right? It depends on the end goal. So I'm thinking about, I've worked a lot with industry, and so I think a lot about the end user. And to me, like, you're not going to be able to access carbon markets with sediment cores. It's too, what's to say that a hurricane is going to sweep that all away next year? And so you need a better idea of how much is accumulating over time as well as how much is in that pool. So that way you can, if there is a hurricane, you can validate in the MRV process, right? So I think you got to do everything, really, is what it comes down to.
Great presentation, Graham. Really liked it. Also liked that Abrams River was on there too, close to home. So I just had a quick question about, like, you were talking about like the Coastal Protection Act and things going on in the maritime provinces. And I'm just thinking about this from like a community or like private perspective, especially when it comes to like accessing carbon markets. How is a municipality going to like actualize this information, do you think? And what are like some of the like really high risks for them? Because I think you just mentioned one, like for permanence, especially for like a voluntary carbon standard. Yeah.
So, I mean, the politics in Nova Scotia, especially right now, are there's been a big backlash to coastal restoration recently through, I would call a misinformation campaign, but
Me too.
there's some stakeholders that have interests that have definitely warped the conversation, and I think broad education is the first step there. And so Nova Scotia is one of the reasons I'm working in New Brunswick, because Nova Scotia is a whole different beast right now politically. But I think from the community, if the community is interested in breaching dikes and restoring, let's say, marsh, low and high marsh system, then in theory, we're not there yet, but if you have a good model of how the carbon accumulates over time, then you can say that's what it is. You write that into your report. Then you say how you're going to monitor that over the next 20 years. And that's the validation component. If a storm comes through and wipes it all away, then you're no longer that's no longer like That's built into the carbon market. It's the same way that if you had a forest that burned down, but the forest was certified carbon, there's mechanisms to deal with those events, but you need to know how much carbon is moving where before you can even have that conversation of building that in the first place.
Yeah, thank you.
Any other questions?
Good time. Okay.
This will be, if you guys have more questions for Graham after this one, move on. Come to the social and ask Graham questions at the social.
Okay.
There you go.
A quick question. Do you plan to use tracers like radium or radon for measuring your lateral fluxes?
We've talked about potentially doing that, or maybe dyes with the groundwater movement, but we haven't come to any conclusions about what the best plans are. Yeah. So, good question. Ask me again next year.
Well, thank you very much for your talk, Graham.
Our next speaker is just me. I'll be talking about the ecosystem evolution of a constructed upland during its first 6 years of development. And we're really just going to focus on its ecosystem function from a carbon perspective. And so before we dive into the constructed system itself, I think it's worth giving a little nod to the boreal ecozone. And I don't need to preach too much to this room about the importance about the ecosystem services that the boreal ecozone provides. But one of the main ones really is that sequestration and storage of carbon in both the above and below ground components of this ecosystem, which is really comprised of open water areas, wetlands, and these forested uplands. And so here on this, will this laser work? All right, on the on the left-hand side here is just a map showing the overall carbon sequestration capacity of the land surface, and you can really see that the boreal has a large amount of that land, that carbon sequestration capacity. On the right-hand side there, we're just zoomed in to the region of Fort McMurray. And here we're really just highlighting that change in that CO2 sequestration capacity when there's a change at the land surface. And so whether that is through natural disturbances or through resource extraction. And so Fort McMurray is a region that has a lot of land surface changes that occur due to resource extraction, both logging and the oil sands mines that are up there. And so in recognition of those really important ecosystem services that these boreal landscapes provide, there's been a lot of work put into setting some obligatory legislation in place that requires any disturbed land to be returned to its pre-disturbance capacity. And so in lieu of that, there's been a number of pilot scale projects to look at, are we able to actually return these landscapes to their pre-disturbance functionality? And so this really entails, is it possible to build a landscape from scratch? And when we build that landscape, is it able to actually function the way that a natural landscape would? And so looking at and understanding those ecohydrological processes, especially during that early system evolution, is really important. It gives us insight into, is reclamation possible? But then also it really gives a lot of insight into how can we improve the future design of these systems, especially if we're going to upscale them. And so that's what this work is looking at. It's looking at, how do we capture the functionality of a system that is built? And how does that functionality compare to natural systems? And so how does one measure ecosystem functionality? If I went around this room and I asked each of you to tell me, what does ecosystem function mean to you? I think we would all have different answers depending on what our scope of research is. And so this figure here really just is up here to showcase that interconnectivity of ecosystems and of those biophysical processes. We can see very quickly, it's difficult to concentrate just on, for example, the plant community, because the plant community is dictated by the soil structure and the hydrology of the system. And if there's changes in any one of those components, there will be changes to the overall functionality of that system. And so when it comes to what metrics do we use to measure functionality? It's very difficult to say just measure soil, just measure hydrology, just measure the plants. We really need an approach that kind of encompasses all of these different factors of those biophysical processes. And so there's some really good ecohydrological metrics that we can use for that. Things like looking at the carbon dynamics, evapotranspiration, and if we pair those two together, the overall water use efficiency of these systems. For today's talk, I'm just going to focus on the carbon dynamics because I could talk for probably 4 hours about each of these, and we only have 13 minutes. And so this is our constructed site. This is Lake Mewasin. It's a pilot scale pit lake and it has an adjacent upland watershed. And again, the main goal here is to test out, are we able to build these landscapes? Are they able to function like the natural systems? Are we able to restore some of that pre-disturbance functionality and capacity? And the reason we have that lake paired with that upland is we're trying to mimic that nice mosaic of boreal landscapes that occur throughout the region. And again, where that upland kind of feeds water into the lake, and we have some of that hydrological interconnectivity. And so how has the system developed over time? This is just some imagery and the enhanced vegetative index, really just showing how the greenness of the site has changed over time. You can see that in just those first 6 years, we've had a very rapid change in going from these bare ground conditions to really a widespread plant community that's flourishing and well developed. And that really kind of happened even in year 2. You see that there's really this crop up of that greenness, and kind of from year 3 onwards, you have that widespread plant community present. The timing of plant development, as we can imagine, is really going to help drive some of those carbon dynamics that we see. And so if we take a look at the temporal trends of that development of that vegetation, this is again the enhanced vegetative index. Anything above a 0.2 really represents that green signal of plant community being present. Again, we really clearly see that increasing trend over time of that system developing. And when we compare what we're seeing at Lake Mewasin to a number of natural sites in the region within 60 kilometers of the constructed site, we can see really by year 4, the system has a very similar, both seasonal and the amplitude of the signal is very similar to these natural undisturbed sites, which is really great to see. We have a second constructed upland really close by, only a few kilometers away, to the Lake Mewasin upland, the Nikonoti upland. I'm sure you guys have heard me drone on about it for the past several years. But how does the evolution of Lake Mewasin upland compare to the Nikonoti upland? They're built on similar material. You would really expect that they would have similar evolution. But when we take a look, Nikonoti here is the black line and Lake Mewasin is the green line. You see that there's actually a very different, there's a big difference in the timing of that ecosystem evolution and the timing of that plant signal coming through. And this really has a lot to do with the timing of not just your trees developing, but that understory. And that's really kind of the MVP of the story here is the rate at which an understory comes in is really going to drive and kickstart a lot of those biophysical processes, because the second you have more plants around and they're being established sooner, the sooner some of those larger biophysical processes will kick in. And so how did Nikonoti and Lake Mewasin look during the exact same age? So this is them at year 3. And here you can really see that stark difference between the two sites. And a lot of that is really driven again by that presence and that development of an understory. At Nikonoti, you see there's a lot more of that bare ground, and it's a lot more brushier at Lake Mewasin. And that understory just kind of came through very quickly, rapidly, and really like not a lot of bare ground present. And so when we start to think about the carbon dynamics, we can really start to piece together a story of how a system that might have this bare ground compared to a well-developed understory will have very different respiration and very different photosynthetic capacity. And so when we take a look at the overall carbon partitioning, Here, respiration is in red and our GPP is in green. We can really see those trends that mirror that development of that vegetation quite clearly. If we take a look at the early initial years, years 1 and year 2, especially in year 1 when we had a lot of that bare ground land surface coverage, you see that respiration is obviously the dominant flux here. We just have a little bit of that GPP, but as the system develops and that vegetation comes in, we really start to see that GPP start to take over, start to creep in and start to overtake the respiration rates. And we're slowly starting to enter that territory where over the growing season, we're kind of leaving being a carbon source, and we're slowly starting to develop enough of that carbon sequestration to occur where we're starting to enter that carbon sink territory. However, we can see that by August in both of these years, respiration kind of kicks back up. And this has a lot to do with, again, the understory kind of has already done its work for the season. It's gone through its flowering phase, and we're slowly starting to senesce. And in these years, kind of year 3, 4, and 5, think about the trees at the site. Saplings do take a little bit longer to become more robust and more hardy. And so treed species really take 5 to 10 years to really start to become, to start chugging that carbon properly. And so we can see that kind of by year 6, even in August, that GPP signal really starts to overtake that respiration signal. And this is, again, those trees are now 6 years old, they're a little bigger, they're a little hardier, and so now we have both the trees and the understory kind of doing the work, and we have that real change in that carbon sequestration occurring. We can see this when we take a look at the carbon. This is our net ecosystem exchange. On the x-axis for each year, we have, it's the temporal trend, so each long, skinny part here is a single day. On the y-axis is hour of day. And so here we see both the diurnal trend and the overall seasonal temporal trend. And you can really see that difference, especially between year 1 and year 6, when there was no vegetation, when there is vegetation. And we can really see that system change from being a carbon source to a carbon sink in terms of its NEE. And then we see some variability throughout the years, and this is really now just the system reacting to whatever the hydroclimatic conditions are. And so this is great. I've just told you guys that within a couple of years, a constructed system is able to go from carbon source to carbon sink. Like, everybody wants to hear that. Like, I mean, this system within 3 years was already kind of back to carbon sink status, which is incredible if you really think about it. Like, what have you guys done in 3 years? That's like half the time it took me to do my PhD. So this system did better than I did. But really, those results don't mean a lot unless we have some context for those results. And so this here is a figure that is a summary figure that pulls together. The purple band here is the range of naturally occurring values that we have at undisturbed natural sites of growing season NEE. And then the points on the graph are various post-disturbance sites. And you can see that over time, any site post-disturbance, whether it's reclaimed, post-fire, or post-harvest, tends to take about 5 to 10 years to really start to get into that, back into that sequestration capacity. We do have 2 of the reclaimed sites here. You can see that they kind of were chugging that carbon right away. And if you actually go back into it and you take a look at some field photos and the veg surveys, the same thing happened in those sites that happened at Lake Mewasin, where an understory came through immediately. And that really changes the story, I think. And so that's really kind of a key takeaway here. And where does Lake Mewasin land in this group of post-disturbance landscapes? You can see that it is well on trajectory to kind of falling within that, that purple band, and a little bit of a slow start, but again, the presence of that understory really helped kickstart that process. And so just to summarize, overall, the upland is, evolving and on trajectory to becoming a self-sustaining functional ecosystem. I think it shows some really great progress and some good early signs here, but there are some things to maybe consider, and I think we've learned a lot now from this system, and when we compare it to some of the other constructed systems, this maybe need for a planting prescription that includes a understory as well, maybe a little more deliberately. A lot of the thought goes into what trees are we going to plant and how are the trees going to do? Is there going to be enough water for the trees? But I think the establishment of this understory actually really helps propagate the future longevity of these systems so that the trees have the time to develop and then become a functional system. So that's all for me. I think we have maybe a minute for maybe one question. I want to keep us on schedule here. Oh, thank you.
Sorry, I saw a hand over here first. Hi, thanks. It was really interesting. I'm wondering, was there something specific to the management of the site that had the understory that came in quicker that you have a theory on why that may have happened?
Yeah, it really just kind of has to do with these systems are built, they're basically these large piles of overburden, if you will. And so I think it has a lot to do with what pile they grabbed it from and maybe what some of the original seeds and constituents are kind of left over in that soil. And then I think the other big factor that I kind of talk myself in circles about, the year that you build the system actually also really matters. And so what is the first year of construction and what does your first year post-planting look like? If it is a drier season, I think your system has a much harder time having that initiation. We were lucky here where 2020 was a wetter year. And so I think having ample water in the system really kind of helps propagate that. And so there's kind of multiple things at play here. I don't think any of these systems have had a deliberate understory planting campaign. A lot of more focus goes on to the trees in the system. And then it's kind of what crops up. Sometimes it's a weedy mess. Sometimes, you get some sweet clover and things like that. And so I think it's both things. I think we need to be more deliberate with the understory, but then also, is it a wet year or a dry year will really actually help propagate that. At Nicanoti, we kind of had that where we had kind of 2 or 3 really dry years after the initiation. And I think that actually really also slowed down our ability, the ability for that system to kind of evolve. I say rapidly, but again, 5 years is nothing, or 6 years is nothing when you think about the grand scale of an ecosystem functioning. We're trying to build systems that will, be around for a very long time, hopefully.
Great. Thank you for your question, Kelly, and thank you very much, Nataša.
Yeah, thank you. So up next we have Francis, and we're going to do a little bit of a pivot and talk about some modeling.
Oh, yeah. Thank you. Oh, nice. Oh, yeah. Okie dokie. My name's Francis. I'm going to be talking about the research project I've been working on for the last 6 months with NRCan. My supervisor is Sasha Hararuk, and Derek Sadler is a growth and yield modeler who helped with some of the growth curves here. First, I'm going to give an introduction to the CBM model. Then I'm going to talk about our research goals. I'm going to talk about the methodology we use to come up with our findings. So the CBM model is you don't have to read that whole paragraph there. The CBM model is a forest carbon sequestration estimation model. It has a lot of history. It's kind of an older model, but the reason we're talking about it is because this is the model that Canada uses in order to come up with numbers of carbon sequestration in our forests that we give to the UN for international carbon sequestration reporting. So that's why we're trying to improve the model so that we can have the most accurate carbon estimations to give to the UN there. The way that the carbon model works is that carbon is represented as existing in different pools. So this is a diagram of all the pools in the model. And up in the top left here, foliage, other, and stem wood. This is the model's representation of a tree, just to give an example. So if you imagine a tree, it has leaves, that's the foliage, that all exists in the foliage carbon pool. The other pool is the branches of the tree, and then the stem wood is the actual stem of the tree. Those are the three components of a tree, according to the model. That's where all the carbon exists. As the tree dies, it goes into snag pools. That means it's dead but still standing. When the snags fall over, they become fallen log carbon. And then when they decay, they go into the soil. We also have some input parameters like growth and yield parameters, annual temperature, disturbance history for the site. Those are all input parameters into the model. And the way that the model is actually working is each of these pools is simulated year after year. So it's a yearly time step. The model runs in two different phases. First, there's a spin-up phase. That's the phase where it's trying to come up with a good idea of what the initial carbon is for the site. It does that by iteratively growing a forest and then disturbing it until the carbon kind of reaches a steady state near the end here over thousands of years. After it's reached the steady state, there's one last disturbance. It grows the forest up to the age that you care about in your simulation. And that's when the forward run begins. And that's kind of what we actually care about. That's the simulation phase of the model. So as you can see, it's kind of a shorter time span. It's only in the span of decades rather than millennia. And this is the thing that we're trying to improve in the model. The goal is to try to identify the areas of improvement in the CBM model using decadal carbon change data, because the model's already been calibrated pretty well for carbon stock data. Like for any given year, how can the model do the spin up in order to achieve that level of carbon? It's calibrated pretty well for that. What it's not calibrated as well for is carbon change over time. So over 10 years, does the model do a good job of representing that change in carbon over time? So that's what we're focusing on. And in order to do that, we're using the National Forest Inventory ground plots. This is a great data set of over 1,000 plots across Canada, where experts have gone onto each of these plots. They're all different forest stands, and they've taken measurements of species diversity, site history, biomass. We use the biomass numbers to inform our carbon. And not only have they gone and taken these measurements, in a lot of these sites, they've now come and done the measurements again. So after 10 years and over 600 sites, they've remeasured the sites again. So that's where we get our carbon change data from time 1 to time 2. The way that we do that is we run CBM model using these NFI ground plot sites as input data. Then we calculate comparison pools. The reason we have to do this is because the NFI data NFI program was not invented in order to calibrate CBM. It was invented to measure species diversity, forest ecology, stuff like that. It just so happens that they measure biomass, which we can convert to carbon. So we have to come up with a way to take their biomass numbers and line that up with CBM simulated pools. So we calculate 4 comparison pools to make our comparisons with. Those pools are the above ground biomass, which is living trees, snags, which is standing dead trees, coarse woody debris is fallen dead trees, and forest floor is needles and leaves on the ground that you walk on when you walk through a forest. Then I calculated the incremental change in each of these pools from time 1 to time 2. The main point here is that this is carbon change data. It's not carbon stock data. And then we evaluated the performance of the model by so the black line here is the simulated data. It's continuous. The black points are the measured data. They only measured once every 10 years or so. We took the difference between measured and simulated. That's the red line, that's the bias. And in order to improve the model, we want to reduce the bias. So what we did here was we did statistical models where bias is the predictor variable. And we took a look at all the things that are in the NFI data set. And we said, okay, is bias a function of all of these things? Climate, stand, age, disturbance history, If it is a function of one of those things, it means that it's not properly taken into account by the model. We can take that into account, reduce the bias through that, and improve the model. So I'm going to be talking about bias and error. These are two metrics that I use. They're very similar. Bias is observed minus predicted. Error, I call the absolute value of observed minus predicted. The reason I parcel them out differently is because they kind of show us different things. You see up here, this is someone throwing darts at a dartboard. If they have high error, they're still on average throwing towards the bullseye. There's just a larger deviation of where the darts land. Whereas if they have high bias, the spread is still just as tight. They're just not aiming at the bullseye anymore. So that's kind of the difference between error and bias, which I'm going to talk about here. Three main findings, just to begin with. So first of all, we found that disturbances contributed by far the most to model error across the board. We found that stand density and stand age were major sources of bias in the model. And we found that weather fluctuations were only a minor contributor to model bias. And frankly, we were expecting weather to play a larger part than it did. So here is severe disturbances against above ground biomass error. On the y-axis, there's error and bias. And the way I was looking at disturbance severity here was for each stand in the 10 years that they were measured, if there was ever a disturbance that occurred on that stand, they measured how many trees died and how large of an area the disturbance affected. A disturbance could be anything like a wildfire or a clear-cut road building, insect outbreak, stuff like that. I just multiplied mortality against plot area, and if there were multiple disturbances in a site, I accumulated them to develop the cumulative disturbance severity for that site. So not surprisingly, if there's more disturbances in a site, or I shouldn't say not surprisingly, I mean, this is a good finding that we had. If there are more disturbances in the site, above ground biomass error increases. We also found that bias increases with more severe disturbances. So if you remember, it's observed minus predicted. So a higher bias means that the observed term is higher than the predicted term, which means predictions are lower than observed. So a higher bias means they were under-predicted. The flip side of this is when we look at dead organic matter, dead carbon. Again, more severe disturbances increased model error, but it decreased model bias. So this meant that as there were more severe disturbances, the model overpredicted how much dead carbon there was in the stand. So this is kind of two sides of the same coin. It's basically saying that the CBM model overestimated how many trees died in a disturbance, how much carbon was being shuffled from living to dead carbon pools. We also looked at stand density. So we found that bias was positively correlated with stand density, and stands that were over 1,000 stems per hectare. 1,000 stems per hectare is a fairly sparse stand. So anything over than that was an under prediction by the model. We also found forest floor carbon predictions were less accurate in sparse stands. 1,000 stem per hectare stand would be off in the forest floor carbon pool by 2 tons of carbon per hectare per year. That's quite a lot of carbon. We also found that older stands had higher error and under-predicted snag carbon and coarse woody debris. So a 400-year-old stand's deadwood pools would be underestimated by as much as 0.9 tons of carbon per hectare per year. So the last 3 slides about stand density and stand age, those are probably artifacts of miscalibrated growth curves. So one of the next steps is to calibrate growth curves with for stand age and stand density. We also looked at climate parameters. We actually looked at a lot of climate parameters, but the only one that had interesting effects was CMI, Climate Moisture Index. It gives an idea of how moist a stand was. It's precipitation minus PET. I looked at the cumulative anomaly of the CMI from the long-term average. So a high CMI anomaly of plus 40 millimeters meant that over the last 10 years, a stand on average saw that the CMI was about 4 millimeters higher than the long-term average. So that's a very moist site, and the moister sites were under-predicted by half a ton of carbon per hectare per year. The inverse was true for dry sites. Finally, we saw that some species had some systemic errors associated with them. So again, this is growth curve related. Lodgepole pine and black spruce were consistently being over-predicted by the model. So just major takeaways, disturbances are probably the number one improvement area for the model. That's what I'm going to be working on next. We also found miscalibration of stand age and stand density in our growth curves, and we found that climate was not much of a source as we expected. Thank you very much.
Thanks so much, Francis. That was great. This is really interesting. And we have time for one question.
Thank you very much for the presentation. I teach this in my classes, so it's good to like be able to talk more about this and some of the issues with, all models are wrong, some are useful. I was curious, like I was really interested in the species perspective, which you highlighted. Was there any geographic perspectives as well? Because in CBM, there is like a component to it where you tell it like what kind of eco province or eco zone that you're in. Was there any biases associated with that that you found?
We didn't see any. We looked at that as well, and
Okay.
yeah, across the board they were okay. I mean, those lodgepole pine and black spruce, I mean, those are geographically distributed themselves, so that's
Right.
that's kind of a factor.
Right on. No.
Yeah.
Yeah, just was curious. Thank you very much.
Okay, thank you very much.
Yeah, up next we have Chloe, and she's going to be talking about some dendrochemical analysis.
Yeah.
Wonderful.
Yeah, hi everyone. My name's Chloe Canning. I'm a PhD candidate at the University of Saskatchewan. We're going to do another little shift here, so not talking about carbon, my apologies. But I am going to be talking about some of the first work that I'm doing in my doctoral research, which is really all about looking at how trees are taking up heavy metals and then seeing where those heavy metals go within those tree systems, which was so beautifully explained earlier of stem, other, and foliage. First, I'd like to acknowledge, though, that we are on Treaty 6 territory in the homeland of the Métis, and just that as we're gathering here together today, we are paying respects to the First Nations and Métis ancestors of this place, and we're coming together in the spirit of truth and reconciliation. But this story really starts further north, actually, in Treaty 11 territory. So this photo was taken by my supervisor, Dr. Colin Larocque, and it's taken just outside of Yellowknife. So a few years ago, he was doing some sampling up in the Mackenzie Mountains, and he found himself in the Yellowknife Airport on a layover, waiting to come back to Saskatoon. And he started chatting with an elder who was also waiting to, fly out. And they had their typical, general airport conversations of who are you, what do you do? And he told her about his work as a dendrochronologist and how he basically tries to understand the stories that trees can tell through their tree rings. And the elder had her own story that she wanted to share with him. And her story was that there's a traditional medicine that she would make from the tree bark and sometimes tree sap. And she would make this traditional medicine for anyone who was feeling unwell, and she would give it to them to hopefully make them feel better. However, she no longer feels comfortable making this traditional medicine because she thinks it's actually making the people that she's giving it to sicker. So if you know anything about the history of Yellowknife, you might be aware that there's a legacy gold mine in the area. And when that gold mine was first in operation, it spewed out tons and tons of arsenic trioxide dust. and it covered basically the entire area around Yellowknife and really impacted the community and especially the Yellowknives Dene. And so this is really where my story starts of trying to understand, how are potentially heavy metals coming into contact with these trees, and could they be the reason that elder no longer, or is that why that traditional medicine is no longer being used? So to understand dendrochemistry, which is the field that I'm working in, I just want to briefly cover dendrochronology. So dendrochronology is where we would go out, take multiple different samples from trees. So we might take full cross-sectional discs, or we like to call them cookies, or you might take a tree core. Oh, that's not the pointer. That is, or we might take tree cores. And then we would take those cores or cookies, we would measure them under a microscope. And essentially what we're looking at is we're trying to measure, or we are measuring those ring widths. And we're trying to establish a chronology or a timeline of how that tree has grown. And so what we're looking at, we might pair that tree ring width data with something like precipitation data. So you'd expect to see in a year with high precipitation, our nice big wide ring, and in a year with low precipitation, the opposite, really thin rings. Dendrochemistry uses those same kind of basic principles. However, we're also assuming that when a tree is growing, it's kind of just passively taking up whatever elements are present in its growing area. So whether that's through the soil, through water, or through atmospheric deposition. And kind of the main thing to keep in mind is that we're assuming that these elements are being taken up uniformly throughout this tree system. Now in dendrochemistry, it's more common to just take a tree core and then usually processing these samples through some sort of destructive method like ICP-MS, for example. And so really what we're trying to do is we're trying to create an annual timeline of how certain elements have changed in that environment using these tree rings, right? Because you can get an annual timeline essentially. And really this is what we're looking for. We're trying to see, when a mine opens up, are we seeing increases in certain elements? When environmental regulations come into play, are we seeing decreases? Or for example, if a mine closes, are we seeing changes in elements over time? Now, like I said, in dendrochemistry, it's really common to just look at this one kind of tree core sample, and obviously that's not the full picture of the tree, right? We usually only use these tree cores because when you're going out and sampling, it's basically just common practice, I guess. And if you're doing a destructive method like ICP-MS, you're destructively sampling these. You might have 80 individual samples from that one core, right? If it's 80 years old and you're trying to get an annual resolution. There are now non-destructive methods. So this is X-ray fluorescence spectroscopy data. And again, we're looking at a single core, and you're seeing that these elements these are obviously different elements throughout, and you're seeing that there's some hot spots throughout this one core, but you're not seeing, once again, that full picture. And so really, that's what I'm trying to see. I'm trying to see this full picture of what do elements look like within that entire tree core, because you can get a very different story if it is actually uniform. You can sample those tree cores from any direction, essentially, and you would get the same story or the same picture. However, if it's non-uniform like B over here, if you sample right here, it would look very different, or that would be a very different story than if you sampled in another location. And also, in addition to that X and Y dimension, my research is also looking at how are these elements changing laterally, or vertically, sorry. So also in that Z dimension. So are the elements the same here versus up here? And so to do this, we wanted to establish what do these elements look like in a non-contaminated system. And so that's where these samples come from. Fun fact, I think one of my sites is on fire right now, so that's cool. But we went out to this site that we know have a real long site history of. We sampled 6 native boreal forest species, air dried them, brought them back to the Mystic-Asquin Dendrochronology lab, and then we ended up just cutting them into 5 sections for each of those 6 species. And then the real cool part of this research is that here in Saskatoon, we have the Canadian light source, which is Canada's only synchrotron. And I used a technique called synchrotron X-ray fluorescence, which essentially I'm using the IDEAS beamline at the CLS. And the CLS, for those that may not know, is a particle accelerator. We're accelerating electrons to nearly the speed of light. When we do that, we're creating a range of the electromagnetic spectrum. And so I'm using hard X-rays, putting my samples in front of that beamline, and then essentially when those X-rays hit at certain energies, different elements will fluoresce. And we can detect counts of those specific elements to basically get an idea of what's present and relative concentrations. So this is a semi-quantitative method. So when we're looking at the results, just keep that in mind that these aren't parts per million or anything like that, but it is showing, what is present and relatively that there might be more of an element in one area versus another. So these are the preliminary early results. So what you're seeing is that first, this is just the trembling aspen sample, and you're looking at calcium and zinc, and then sections 1 through 5. So we're going up vertically as you look along the bottom here. And the thing you might notice is that it's not uniform. We're not seeing this perfect concentric rings or ring pattern. We're seeing that there are differences depending on where you're looking. And that's the case both vertically and then also laterally. And so if we continue on, just looking at a few more species here, you can see that a lot of these differences in the trees, they're from physiological changes happening inside the tree. So when we were sampling, we didn't necessarily try to include branches or anything like that. We couldn't see them, right? But now we can see them using our XRF method. And so you can see that when branching occurs, there's a change in the elemental concentrations there. So once again, depending on if you sample over here or you happen to hit part of that branch, you'd be getting a different story. And then you may have noticed in the trembling aspen samples, you can see these inclusions, specifically in the zinc examples here. And those inclusions there's 2 of them in sections 1 and 2, but then the second inclusion disappears in the other sections. And so once again, you're getting a very different picture. We think those inclusions are actually bark, so maybe some sort of scarring that hit that tree. We have to do a little bit more research to actually conclude that. But yeah, again, you're getting a very different story. You might hit those inclusions, you might not. And then finally, the other thing to note is just that obviously when you're doing any kind of heavy metal work or work with elements, you want to limit your contamination as much as possible through whether that's through sampling or processing. Obviously, we cut these with a bandsaw blade, and that introduced these very visible scars. So this isn't revolutionary, but it is actually a non-destructive method, and you're seeing how that contamination is coming into play and how that can change your results. So just going forward in dendrochem research, being really cognizant of how you're sampling and how you're processing your samples. So what does this actually mean for our elder up north? Obviously, you don't need a synchrotron to determine if there's arsenic or any other heavy metal present in environmental samples. But the real potential for dendrochemistry is that timeline effect, right? But what we're seeing from these early results is that depending on where you sample, you're not necessarily getting that same story. And so going forward, I'm doing more research looking at a controlled growth chamber study. And so we're adding different heavy metals in known concentrations in a controlled environment, and we're going to see if those heavy metals are taken up the same way as those these other site. And I'm once again using the CLS to do that. So thank you.
Thanks, Chloe. That was very insightful, and it's going to shape up to be a really great project, I think.
I hope so.
Yeah. I think we have room for one or two questions, if anybody has one for Chloe.
Okay. You gave us a few elements. Are some elements much more variable than others? Are there some which really don't work if you're trying to use dendrochemistry?
Yeah, for sure. So I obviously only showed the ones where you could actually see something. So generally, if there's just calcium and zinc, those are present in biological systems, right, and in cells. So they're going to be a lot more present. When you use synchrotron X-ray fluorescence, like I did, we actually detect 18 different elements all at once. And so, for example, copper couldn't see any of that because naturally there's not necessarily a lot of copper just in the soil, or at least for my samples, there weren't. And so Levi will talk more about copper though in his stuff. But yeah, so some elements definitely do work better, and previous literature would also show that. Any other?
I was curious, why is there a higher relative proportion of heavy metals in the bark, it looked like?
Yeah, so we think it's because, well, yeah, that's where the phloem is, and so that's where the tree is taking up all of those nutrients and whatnot. And then we're seeing that it's not being like distributed into the rest of the xylem wood, or there's no heartwood in these samples, but yeah, so that's why, I think.
Great. Thank you, Chloe. I think that's all the time we have.
This brings us to our last talk of this session, which will be a great talk.
Absolutely.
Absolutely. Levi is going to keep talking about the synchrotron. I love this. This is great. Great content for us. So take it away, Levi.
All right. Yeah, thanks everyone for sticking around to the bitter end. I've been talking to the organizers and they said they intentionally saved the best for the last. So I'm a PhD student at the University of Saskatchewan here in the Department of Soil Science, and I'll be talking to you about mine tailings, mostly about metals, but don't worry, there's a bit of carbon at the end. So there's a reason they let me in the door. Before I start, I want to acknowledge that I live and work here in Saskatoon, so that's Treaty 6 territory and homeland of the Métis as well. Many of the pictures you're seeing, the work that's been done, the samples are coming from Treaty 9 territory. There we go. So this is the type of system that I'm interested in. These are metal mine tailings. And so they're kind of an environmental problem in that they contain a lot of heavy metals of varying types and concentrations. And these tend to be spread across the boreal forest in Canada. There's lots of mining going on there. And I'm really interested in what happens when we try and restore these sites. What that'll look like generally, it falls into kind of two main categories. One is if the mine has money, then they will be able to put some kind of substrate on top of this site, prevent it from oxidizing or leaching. Then they can revegetate that, ideally with native boreal plants, and that will chemically and physically kind of stabilize the site. The other option is in some of these places where resources are more limited, they will then either minimally amend them, say with lime to adjust the pH, and then allow revegetation to occur. Or if the site has been abandoned for a long time, natural revegetation may just happen and trees start growing in it. And so in all of these situations, understanding how boreal plants interact with metals and tailings is very important. And so as a good soil scientist, if I hear that kind of thing going on, my first instinct is to want to do a soil test of some kind. And so that's what I'm going to talk to you today about. This is kind of the first step of my research, which is to get a handle on the variability of a relatively fresh mine tailing cell with respect to elemental concentrations as well as speciation. So the pictures you've been seeing come from my research site. This is a relatively new gold mine tailing cell from Northern Ontario. It was closed about 2 years ago. It's got a mixture of ores going into these tailings, mildly acidic pH of about 6 at the moment, and the two metals I'll talk to you most about are copper and chromium today. So to start off, we were able to take some samples from varying locations and depths, really trying to chase down the hypotheses that there may be some spatial variability, both between sampling points as well as between depths. So you're seeing on the x-axis here, I've got chromium on the left and copper on the right. Those are just arbitrary names given to different sample locations. And then from top to bottom, you're seeing increasing depth from the surface. So the 0 to 15 centimeter fraction, then the 15 to 30, and so on and so forth. And those were all compared separately to avoid any kind of pseudo-replication. And the story here really, that vertical redistribution that I assume might be happening, either leaching downward or evapotranspiration upward, was just not occurring. At least I wasn't able to capture that in my sampling. However, between locations, you can see there are some significant changes at most depths of elemental concentrations, roughly following the same pattern for both of these metals. However, nothing super crazy, everything sticking in that kind of hundreds to low thousands of ppm, not changing by orders of magnitude. This is not the most interesting part of this for me, though, because as I've said, I'm interested in plants in these systems, and so that really will, things like bioavailability become much more important. And so I'm lucky to work next door, essentially, to Canada's only synchrotron, where we're able to directly measure things like speciation, which have direct controls on that bioavailability. And so we'll get into the first technique that I've used, which is called ZANES, because everyone at a synchrotron loves their acronyms. It stands for X-ray absorption near edge spectroscopy. That's not terrifically important, but it's fun to say. And what that looks like, this is a chromium ZANE spectra, and then it's been fit with a technique called linear combination fitting. So in blue there, I have the measured experimental data, which is necessarily messy. It's got chromium of a variety of species in it. And then in red is the linear combination fit model of that data, and it's comprised of everything underneath it. And so in this instance, only two phases going into that. That's chromite at about 80%, and then chromium nitrate at about 20%, making up that fit. To mention, those are measured as well at the same time as your environmental samples. So you have your pure reference compounds that you then plug into some software, and it tries to approximate that messy environmental data. Of note here, though, that red line and blue line don't match up perfectly, so that indicates I'm likely missing some kind of species, and I'll get back to that in a second. However, I'm still looking at that hypothesis of, is there any kind of spatial variability in terms of speciation here? So on that right in the figure, everything except that white line at the very bottom are just different chromium samples from various locations and depths, and you can see that they don't change very much. Barely at all. So roughly that 80 to 20% ratio of chromite to chromium nitrate holds true for all of these samples. Interestingly, though, that white line at the bottom is a standard. That's potassium chromate, so that's hexavalent chromium. And it's got that very intense pre-edge feature, it's called, right there, which is indicative of that, again, that hexavalent chromium. And so just luckily for this mine and the environment generally, it's not in these tailings. However, I said I'm missing a standard, and so you guys have heard about this technique before. This is XRF imaging that Chloe has talked about before me. I've just applied it to tailings instead of trees. And so you're seeing here tailings, a false color image with iron in red and chromium in kind of that white-blue color. And so by looking at some of the places of co-location, I can get an idea of what may be going on chemically. So then pulling apart those three different spots of interest, you can see the image on the left and then on the right, that's just the correlation coefficient at each spot, trying to pull that out. A and B there have a very bright chromium signal on top of a very bright iron signal that you can't see underneath it in this image. But you can see the correlation is very high at those locations as well as that slope. And so indicating one of those is likely that chromite ore phase, which is a chromium iron ore. But more interestingly, you can see here at C, there's a much more diffuse chromium signal there sitting on top of that larger iron particle. However, the correlation at location C is still very high. And so what this seems to indicate to me is possibly chromium has dissolved in some way and then either co-precipitated with iron or reabsorbed onto iron. And so that's likely my missing phase. As well, this would be a more bioavailable form of chromium, so something to keep an eye on in terms of this cycling in the environment. I also have some copper that I want to talk to you about. And so luckily for keeping this presentation interesting, there's a lot more variability here. So you've seen one of these before. This is Zane's data again with the linear combination fit. The shape is a little bit different, but the bones are the same. So that blue line at the very top is that measured experimental data, so tailings at the 0 to 15 centimeter fraction. And then red is that linear combination fit, and everything underneath it is contributing to that fit. So in green, you'll see that's chalcopyrite, so a copper, iron, sulfur ore, contributing about 40% of the fit in this sample. And then in blue at the very bottom is a copper oxide, very negligible part of this. And then, now's the carbon part, in purple and yellow, those are copper adsorbed to various organic acids, so humic and fulvic acid. And in this sample, they're contributing about 50% of the fit. And this is pretty interesting because if you think back to those pictures, this site looks a lot more like the moon than anything else, so not somewhere you'd expect a lot of organic carbon to be. And this is true, in fact, I haven't measured anything more than about 0.02% organic carbon. However, it seems to be playing an important role in copper chemistry here. So I've looked at, I want to talk about some more locations. So these are at the same depth, that 0 to 15 centimeter surface fraction, just at a different location on the tailing cell. So again, chalcopyrite showing up in a high percentage, about 70%. We have roughly 10% copper adsorbed to fulvic acid, and then some copper sulfate and actually some elemental copper showing up in this fit. And then at yet another location on the tailing cell, chalcopyrite about 60%, and then we have roughly a 30% contribution of those copper adsorbed to organic acids. So just putting that all together here, chalcopyrite is in black, and then orange and blue are that copper organic acid association. You can just see at these 3 sample locations, there is some variability for sure going on in terms of copper speciation. As well, I've been really trying to make that vertical redistribution or variability be a thing, and in copper, actually it is when you look at speciation. And so I was able to take some of those profile samples down to about 1 meter. You've seen this one before. This is one of those 0 to 15 centimeter samples. I was able to then take in that exact same auger location, just down to deeper depths, and do that same speciation work. And so you can see here that chalcopyrite contribution goes up to about 80%. We have roughly 10% copper adsorbed to organic acids and a little bit of copper sulfate. And then just past 1 meter in depth, chalcopyrite is dominating this fit entirely, about 90%, and then the rest being copper adsorbed to humic acid. So again, putting that all together, you're seeing that from left, that's the sample closest to the surface, 0 to 15, going down to the far right, just past 1 meter. Chalcopyrite, again, in black, increasing near depth or decreasing near the surface, whichever way you want to think about it. And then in orange and blue, again, that copper adsorbed to organic acids. So altogether, really the variability that I was able to capture, there's limited spatial variation in terms of those absolute elemental concentrations for copper and chromium, much more so between locations than between depths. Unsurprisingly to everyone, I think, not all elements react along the same timeline or according to the same factors. So you'll remember that chromium really didn't change no matter where the sample came from. However, the copper had a lot more variability. And then, interestingly, copper is very controlled by organic material in the system, despite there being such a low concentration. And so this is a relationship that we know from soil, actually. There's a high affinity for copper and organic material in soils. But seeing it show up in this very contrived system as well with such low amounts of organic carbon is very interesting. And then finally, copper appears to be moving very swiftly into association with organic carbon. If it's not in that chalcopyrite phase, there's very low amounts of it in terms of an inorganic copper salt, like copper sulfate or oxide, the second most dominant species in the system is that copper organic association. And so this is helpful for understanding this tailing cell, but as well similar chemistry sites, the things that may be affecting variability as we close some of these tailing cells in the boreal forest. With that, I'll leave up this slide with some people who've been very helpful to my research, and I'll take any questions if you may have them.
Thank you very much for the presentation. Amazing work and really important, I'm sure. So the copper association with the humic and fulvic acids suggests that it is associated with biological activity. But is it, do you think, a passive association? So you have pedogenic processes forming the acids, and then the copper just, I guess, hitches a ride? Or is it rather some sort of microbiological mediation there that, do you have any sense of that?
I'm a bit biased. I'm an inorganic soil chemist, or at least my supervisor is, and I'm getting there. But yeah, when we first saw this, I was pretty skeptical of it because I looked at those tailings. I'm like, there's no way. But we got in contact with the mine, and they're actually, the process that they're using is a carbon in pulp recovery. And so they cyanidize it, extract all the gold, and then that gets absorbed onto a carbon pulp, which then they, in theory, are recovering 100% of that, but there's always some loss. And so either that's where this carbon is coming from, so it's inorganic, at least to start with, as well some of their water is possibly coming from outdoor sources that have some organic material in the water before it gets out there. And so those are the two places we're assuming it's coming from. In terms of biological activity, there have been a couple of samples that I extracted for pH and things like that, and then left over the Christmas break, and then they got pretty stinky afterwards. So I think there's something in there, but I haven't done any of that microbiology stuff. Yeah.
Any other questions for Levi?
It's a very nice day outside. Thank you.
All right, everyone, that brings us to the end of the session. Thank you for coming out and supporting our ECRs. Please come out at 5:30. We are heading down to Cathedral Social Hall. We'll have a little social, have a couple of drinks. We can continue the conversation or have other conversations. And there will be a little bit of food provided by the BGS. So come on out. It's going to be a great time. A nice way to kick off and end the first day. If you do head to the AGM, don't worry. We will still be there even after the AGM finishes, all right? So thanks for coming, yes?
Before we head out to the bar, there's a friend from the conference happening at 5:20, and it starts in a few minutes. If we can go and support our...
Yes, of course, of course. Would you like me to repeat that over the, did everybody hear that, or? I think at 5:20, we have a Francophone meeting as well, so head over there first, then head to the AGM, then come for beers. I'll be there the whole time, so you guys just come by and see me, and we can talk about anything you want. All right, I hope everyone had a really good first day at CGU, and let's have a really good week, all right?